I was premature. Wendy, did you have something to say?
Oh, just that I would help getting the NC site if we could only do it in the first or second week of May. I'm not available at the end of April, but I could help. And I can ask NC if it's available the first or second weekend in May. So
Okay,
I'll just put it
that's
out there.
great.
That's the first step, getting a place.
That's great. I was kind of had that in mind anyway, so
Okay.
wonderful. Okay. Anu, if you would introduce our speaker.
Hello, everyone. It's a great pleasure to welcome LiAnn Noonan to our program today. LiAnn is a graduate student at Western Washington University, where I teach as well. She's originally from Oahu in Hawaii. She went to Western, though, as she was an undergraduate at Western. She graduated with a biology degree in 2019. She says she went home and dabbled in marine biology for a while. One can understand why, with the enormous potential of the marine world in her stomping grounds at home. But she says her heart was really set on plants, in particular, herbaria and their use in botanical research, especially in systematic botany. So that's the modern branch of what used to be called taxonomy, modern branch of biology, which seeks to classify plants and understand their relationships to each other from the evolutionary perspective. So systematic botany is of great interest to her. So she came back to Western, and she is now a graduate student, and she'll describe her research project. Very exciting and very interesting stories. Her research is guided by Mike Williams. Some of you may remember Mike Williams. Dr. Williams presented a talk to us a few years back. I can't remember exactly, more than 2 years, pre-COVID days. And he talked about a dune grass in the Aleutian Islands. And that was a marvelous story because it was part travelogue, part World War II history in Alaska, in the Aleutian Islands in particular. So Mike Williams is guiding LiAnn's research, and she'll tell you that whole story. So over to you, LiAnn.
Thanks for that introduction, Anu. I'm just going to take a second to share my screen, so hopefully everyone will be able to see my PowerPoint. Give me 1 second. Let's see. All right. I'm just going to move you guys all to the bottom. If there are any questions, hopefully we can just save that for after. That would be wonderful. So I'm going to resume my share. Can you guys see my screen? Awesome. All right, wonderful. I'll go ahead and get started then. So good evening. My name is LiAnn, and today I'm going to be presenting my proposal, which is titled Continuity of Floristics in the Alpine Zone of the Cascade Sierran Axis. And so in all, what I'm presenting on is I will be identifying whether flora are capable of migration by comparing historical and current presence absence data through the use of herbaria. Oops. Oh, it's not working. There we go. All right, so really briefly, an overview of today's presentation. I will be discussing some background information first on alpine plants and potential responses of alpine plants to climate change. I will be introducing my question and hypotheses, walk through some of my methods, which is kind of divided into gathering baseline data and analyzing the focal taxa data. And then finally, I will be discussing the importance of the project, its associated questions, hypotheses, and how I plan on executing the study. So let's get to it. So climate change has resulted in shifts in climate zones, which is especially visible as we increase in latitude. So according to the IPCC's special report on climate change, these trends are projected to result in mid to high latitudinal habitat shifts towards the poles. And so as you can see here, let me grab a little laser pointer. As you can see here, we sit in mid latitude within the northern hemisphere. So we will see temperatures increase more poleward, and therefore we expect the niches to also be shifting along a similar trend. And so in addition to the northward movement of the available niches, we will also expect to see species shift towards higher elevations. And so I just used some basic icons over here, but the icon right here represents the conical shape of most high elevation habitats, while the yellow would represent the baseline of historically recorded alpine zones. And then the red lines indicate the surface area that we're going to be losing as we increase in elevation. And so elevation, therefore, is going to be continuing its upslope range contraction until there's essentially no longer enough habitat left to support a majority of species. So what then does this mean for endemic alpine flora, particularly within alpine habitats? And just first off, what are flora for people that might not be as well versed in this? So flora are plants that are found within a specific region, habitat, and or geological period that can essentially be used to assess the biodiversity of an ecosystem, especially if endemic, which leads me to the next term, endemic. So endemic species are essentially found within a specific region and nowhere else in the world. And they're extremely important in conservation biology because they depend on management to persist in an area that they're restricted to, particularly nowadays. And so this restriction is due to historical factors, which might explain how taxa became restricted, and also ecological factors, which explain the current distributional limitations. And so endemism, particularly in my study, is important to define because there are pretty broad applications of the term. And so an example of why stating my level of endemism would be important would be, for example, stating that a species is endemic to North America, which is pretty broad. And this is vastly different from being endemic to the West Coast, the state of California, or even down to Tuolumne County. So my study will be restricting the definition of an endemic to the state of California, which again is represented by that black circle. And that's in order to align with the California Native Plant Society's definition. And so that is actually where I sourced my taxa, and I'll get into that a little bit later. But my study essentially is going to be accepting species that are endemic to these specific habitats within the state, but no larger than the state of California. So if it crosses over into Nevada, then that means that no, that is not an endemic by my definition. So now that we've covered a few important terms, what are the possible ways that alpine species might respond to the loss of higher elevation habitats? So here's a really short list of 5 potential responses to climate change. So first off, there is acclimatization. So alpine species could potentially tolerate or acclimatize to changes in climate. However, alpine endemics are adapted to a very, very narrow set of local conditions within the microenvironment. And so they might have a reduced ability to respond to these environmental changes. Another potential response would be adaptation. And so this is also an unlikely scenario because adaptations may occur rapidly in certain species like bacteria. However, changes in climate at higher elevations are occurring much quicker than slow growing alpine species may be able to adapt to. Populations may then either be severely reduced or driven to extinction, both of which are worst case scenarios and reflect the failure of species to acclimatize, adapt, shift upslope, and or migrate. Which brings me to my final and most probable potential response and the focus of this study, migration. So a study was conducted in 1991 by Brian Huntley on how plants respond to climate change. And he essentially found that some, but not all plants will follow habitats that meet their specific niches. Therefore, by identifying endemic alpine flora's current distributions, we should be able to track potential migrations using historical data. However, Huntley's study, along with many others, are not specific to endemic alpine flora, and they tend to focus on lower elevational flora using either phylogenetics, floral checklists, or recent collections, which now brings me to the purpose of my study. And so the purpose is that we will be focusing on the alpine regions again of the Sierra and Cascade Axis. And we aim to ultimately determine determine the distribution of alpine endemic flora based on their indices of similarity, and then determine whether the historical populations have indeed shifted over time based on those indices. So in order to justify my questions and hypotheses, I do need to discuss some more background information on the methods before diving straight into what my summary was primarily composed of, and also my methods and analyses. So continuing on to some more background information, why are alpine zones, specifically in the Sierra Nevada and Cascades, really great for studying the effect of climate change on flora? And it's because they're generally unaltered by humans. And this is because higher elevational regions like alpine zones are really isolated compared to lower elevations. And therefore they're not as heavily affected by things like habitat conversion, fragmentation, or most human population pressures directly, at least. However, this doesn't necessarily reduce the effects of climate change on these alpine regions. They're also, therefore, extremely sensitive to climate change. And as previously discussed, climate change may result in the reduction of that upslope area, leaving the options to either be reduced, become extinct, or migrate. And this means that they essentially provide us with an as close as we can get to an ideal natural lab setting to study the effects of climate change. And again, primarily because direct human impacts aren't heavily present. And lastly, they are a great biodiversity hotspot. So according to a survey that was performed in 1996 by the US Forest Services, the Sierra Nevada represents approximately 20% of the land cover of the state of California. And so you can see the Sierras here in blue. But the Sierra also hosts over 50% of the state's flora, where approximately 405 or 15% of these species are endemic. So what that means is that this blue region hosts the same amount of endemic flora as all of the other colored regions collectively within California. So that's a very high amount of endemism in a very small strip. And again, this is thanks to their specific habitat requirements. However, this also means that climate change has the potential to greatly reduce our planet's biodiversity should the projected warming trends reduce the habitat continuously within the Sierra Nevada. So within the Sierra Nevada and the Southern Cascades, I selected counties on the basis that they were in the general Alpine Zone of the Sierra, where if you see here, it's really small. The green indicates elevations between 2,700 to approximately 3,900 meters, which is the general Alpine region of the Sierra Nevadas, according to an analysis conducted by Wendling in 2011. The blue, on the other hand, indicates elevations between 1,500 to 2,700 meters, which are areas that are likely going to be containing those migrating flora. The 13 counties that I selected, which you can kind of see outlined here in black, I couldn't blow up it any further, unfortunately, but there are 13 counties here. They follow essentially that northward distribution of species, and it overlaps into the Cascades right up here in the Siskiyou, Modoc, Shasta, Lawson area. And this is important to note because the Cascades are quite young and they're also active while the Sierras are not. So substrate that the plants will have access to will differ in habitat between the two ranges. So when defining the Alpine Zone, I kind of ran into a chicken egg scenario. Do I determine the Alpine Zone and define that before first determining what endemic alpine flora are and how they comprise of that area? But I ended up selecting the flora first because they came predefined by the California Native Plant Society as alpine or not, but I will discuss that in the next slide. So before that, I'll just get into defining how I got my Alpine Zone. And so back to defining the zones, Alpine zones are not ubiquitous. And so that means that one set definition can't be applied to evaluate every alpine zone at every latitude. So categorizing flora into an area that's not really unanimously defined can be quite difficult. And so a few ways that alpine zones have been identified in the past are through things like setting tree lines as lower elevational limits. And so you can kind of see the tree line right here, but this is what I will be using as my cutoff. So I kind of box that in red. Two, they could also be using set elevations. So I believe 2,700 meters is the average across the Sierras. And then three, they could also be using other characteristics of the habitat for the specified surrounding flora. So the flora that are considered centered around the Alpine Zone from the California Native Plant Society was recorded. And so I will talk about that in the next slide again, but their elevations are essentially going to be used as an average to calculate the bounds for the study. So that will be the general Alpine Zone because it's within a more ubiquitous area. I'm still working through amending my data. So this step has yet to be completed, unfortunately, as we will see going throughout. COVID did unfortunately impact my study quite a bit more than I was hoping it would. So there were a few setbacks, but getting back into things, I found my focus taxa through the California Native Plant Society inventory of rare and endangered vascular plants. And you can see that right here. Also got the beautiful tabbed copy of Chaos that I ended up using for my study. And I used an older edition because there were likely plant populations present that align with older collections since it was published in 1994. So after I selected the book that I was using, I went through each flora in the inventory and I tabbed and selected every flora that was present either in the alpine boulder rock fields or alpine dwarf scrub, plus or minus distributions in subalpine conifer forests. And I say plus or minus subalpine conifer forests because as long as flora were centered within the alpine zone that I selected, a few lower elevation distributions were completely acceptable. And so once the habitats were recorded and my flora were selected, I reviewed my list and then I removed all species that were not listed as endemic because again, I want them to be within only California. So once the focal taxa were selected, I started gathering data available from digitized herbaria. And again, I'm using open access data because COVID did prevent me from going out in the field and actually doing another project that I had planned on doing. So this was our contingency. And to clarify, herbariums are not a place where herbs are grown, for those of you that aren't familiar with this. Herbariums are essentially libraries that are full of preserved, systematically arranged and identified plant species that essentially document historical and current plant diversity. And so usually the plants are flattened in a press, dried, and then mounted on archival paper where a label is then added. And so I kind of zoomed in on one of the boxes here that you can see. And so this one has been really nicely preserved. We do like to splay it in a way that you can see the reproductive structures and things like that, other things that would help you identify what species you're looking at. And then directly below that, I have an image taken from Neil Kramer's site. And so that is what the species looked like when it was alive. And so the other piece that I wanted to focus on were labels for herbaria. And so the labels here are usually located at the bottom, someplace off to the side that everyone can see. It's really clearly annotated. And so they usually indicate the location, for example, where the plant was growing, the collector, date, and there's also a bunch of additional information that might help people reference that plant and anything that may have been associated with it. And so the additional information that is documented kind of regards things like the appearance of the plant, the context, or the habitat of where it's found, to sum it up. And so each herbarium specimen here was assigned a unique number, and then it essentially functions like the catalog number of a book in a library. And so essentially a herbarium is a library of dead plants, but a very useful library, something that can frequently reference and is great in terms of studying things historically. So I gathered my data from the Consortium of California Herbaria 2, which is the current working portal. And the data was sent here from multiple herbariums, which is why it's called a herbaria. And I filled out the taxonomic criteria essentially when going to find all of my species after I sorted them in this wonderful book. And I just simply added the name here. It is a little bit technical with whether or not you use subspecies, variant, things like that. And so it took a little bit of time to try and actually find what I was looking for. But once the search was complete, a list of records associated with the herbaria then appears. So I'd hit enter, and this would be a list that's brought up, for example, for this species. And so all of the records should essentially have a list with the specimen number, collection number, collection date, county, elevation, lat, and long, with the latter being what I really focus on. And older records might only have relative site descriptions or township and range surveying systems, which I'm sure a lot of you are familiar with, is the old way to read a map. And so I will be assessing all of that a little bit later. Again, still parsing through my data, but I'll essentially be looking at lots of that later on. So hopefully not everything will need conversions from the range and township, but it looks like for right now, we do have a lot of lat long, which is great. If not, I will definitely be resourcing Mike Williams to help me with that. But some of the records here may have been imaged and some of them may have not been. And so that kind of presented a bit of a problem because they were lacking in a little bit of detail. So it could have been, for example, like the latitude and longitude, just like the locality, the elevation, descriptions, and other things like that. And so if a record was incomplete, I did have to request a new image from the herbaria. And so this is an example of what a complete image would look like. It's the previous image that you guys saw, and it had everything that I specifically needed for my study. And if they didn't have it and this information was not filled out here properly, I did have to request an additional image. So after running my list of focus taxa through the CCH2 herbaria or the database, I noticed there were quite a few gaps in many of the records, particularly in species that should have been present within the Alpine Zone, especially because they weren't necessarily classified as rare. And so this led me to two options. So first, I could again contact the herbaria and identify whether there are undigitized collections, or two, find other potential sources of data. And I heard you guys mentioning earlier that you guys were using inaturalist to find spots or localities for these cones, which was awesome. And so that's actually what I started doing. So focusing on the latter, I started compiling research grade records of species from inaturalist that document key features which align with the digitized records. So that means, again, I want the latitude, longitude, date of collection or imaging, and blanking really quickly, and the elevation. And so I actually did end up finding records on inaturalist that were not present in herbaria. So that was really great. And it gave me hope that, you know, maybe herbaria do have a lot of undigitized records. And so following up on that second option, I actually did send out a bunch of emails this summer to herbaria such as Cal Academy, Chico State, UNLV, and UCLA, just asking if they had any undigitized records available. And I did hear back from quite a few of them, and I actually gained access to a few restricted data sets as well. So that was really great. And so that's essentially what I was doing all summer, was just correspondence, trying to get some data and also parsing through the data. And so a few reasons that these records may not have yet been digitized is that this is still a fairly new method of releasing information to the public. So all of the records just might not have yet been pulled from the shelves and manually entered in. And so I actually told the classmate a few quarters back that they had just finalized the CCH2 herbaria because the quarter before I was working out a CCH1, and that's had a significantly smaller amount of species available. So that just kind of goes to show how recently this open source method has been developed. Additionally, imaging can be kind of expensive, and volunteering is pretty limited and might not be prioritized, particularly in times of COVID. That gets a little bit messy. So this summer, again, was primarily spent just trying to contact the herbaria, see if there was any data not yet published, and then manually correct the imported data sets because they didn't always align with the herbaria labels. So the metric system might have been off. Where it should have been meters, it was feet. And so just going through a list of essentially 5,000 samples was what I was doing during the summer and what I still am doing. So Once all of my data has been gathered, I'm gonna start moving into what I hopefully will be doing in this upcoming quarter. I will be using a package called Let's R, which is created by Bruno Villela and Fabrizio Villalobos. It's fairly new, to transform the spatial data of species into a presence absence grid. There's a lot of GIS work that's going into that I still haven't been able to figure out yet. So if any of you are really well versed in GIS, that'd be amazing to talk to you guys after. And again, I haven't quite finished amending the data yet, so I haven't really had an opportunity to try this package, but it's pretty short, and hopefully I will be able to get through that again within the next quarter. So after my data has been transformed, I can start focusing on my questions. And so I'll be first addressing my question of whether populations are similar. And to do so, I will likely be running a Sorensen's index of similarity, which focuses on the presence or absence of species, where my null identifies that populations are all distinct and migrations are not occurring. So that means that if I reject the null, then populations are likely unable to migrate. And if I fail to reject the null, then I will move forward with analyzing whether populations have shifted over time, because the populations were deemed continuous over an area, which is kind of backwards. And again, I have a very basal background in stats. I'm actually taking multivariate right now. So if anyone has further recommendations for analyses, I'd be really grateful for the contribution. And then if I was able to gather my similarity index, I can after then determine whether historical populations have shifted. And should no historical taxa have shifted, it likely means that the endemic flora's response to climate change is anything but migration. And to address this hypothesis, I was thinking of running a multivariate analysis of sort, but I really haven't gotten far enough with collecting or even looking at the indices to get a good grasp on what I should finally be using. So with that, I'd like to thank my advisors, Dr. Ben Miner and Dr. Mike Williams, for all the support that they've provided me during these really crazy times. My committee members, Sean Belling and Matt Zinkrath, for taking the time to provide feedback and support me futurely in my ArcGIS and stats endeavors. The Fraser Hodgson families for funding part of my research this summer, and Anu for inviting me to present, and again, all of you guys for listening. So thank you so much for having me. Questions? Oops, I will stop sharing.
For questions, let's, Denise, can we move everybody to a panelist? How do we get people to talk?
Hi, do you want to do that now for
Yeah.
questions?
Yes, please.
All right, just a moment. I'll just go through the list and do that really quickly for you.
Right.
Welcome in,
So-
everyone. You're welcome to turn on your mic and your camera when you get your panelist invite, and that will bring you into the room with LiAnn and Anu. Thank you for
Right.
joining.
So feel free to simply ask your question, anybody who's ready.
Oh, Eric.
All right, yeah.
Hi there. I wonder what you can you speculate on what you might find? Do you have any sense of the results at all? And what can you tell us about past research into plant migrations under climate change?
Yeah, so just based on looking at the data alone, I haven't been able to see any sort of pattern, particularly because my spatial scale is just so large that I still need to divide up the land masses into grids. And then based on where things are located within those specific spatial grids, that's the one way I'll actually be able to tell whether or not something was similar to each other and has actually moved over time. Just because if I were to have compared, for example, Shasta and Tuolumne, those land masses are very different in size. And so that wouldn't really be a reasonable comparison, unfortunately. In terms of past research, There has been quite a bit of work done specifically with like phylogenetics, which is something that's at least Mike and one of my former advisors, Eric Deshane, had asked me to do. But specifically, I'm not well versed in that. And so we leaned more towards like the open access data, particularly because of COVID. And so there aren't too many papers out there that have just strictly used herbaria, to my knowledge, specifically looking at endemics in higher altitudes or elevations. They're more so focused on lower dispersals. Did that answer your question? Mark.
Yeah, I'm curious, two things. One, why you chose to work with California data when you're doing a degree at Western. And two, I've looked at a lot of herbarium records, and there are some that go back, 1900 thereabouts. And I'm wondering if, the records from down there go back far enough that give you a long enough time scale to determine whether things are, really moving.
Yeah, and it's actually been really wild to see plants that are 200 years old on paper. So definitely, yes, we've gotten species from the 1800s, which is absolutely astounding. So I'm not too worried about the time. What I'm really, really worried about is my sample size right now, just because having those collections available from such a long time ago might be a bit threatening to the statistics that I'm using. Although the time frame, I'm not horribly worried about. I'm really excited to actually dive into that. Granted, the sample sizes are large enough. So I might end up having to eliminate a few of the 64 species that I currently have because of that lower sample size. And then, sorry, could you repeat your other question for me really quickly?
I was wondering why California instead of, Washington, instead of the Cascades, for example, Washington and Oregon.
Yeah, totally. So I did initially want to work with plants in Washington, of course, that's why I'm here. And so I was originally supposed to work with the SAM project. I believe Eric Deshane gave a talk on that about 2-ish years ago now to this day, which was amazing. But unfortunately that fell through. And when I was looking at the open access data that we had here, it wasn't necessarily alpine enough for me to consider looking at. And also the data that has been published in California was substantially larger than the amount that was made available here, which is unfortunate because I know we have amazing herbaria here that just don't have the data quite yet up online. So did that answer your question?
So is there a field work component to what you're doing, or is this all looking at herbarium specimens, remotely, more or less?
It's all remote. Unfortunately, I wish there was a component where I could go out and do some field work. I had actually planned on doing that last summer. So I would have been traveling to the herbaria to at least aid them in getting more images up quicker, and or being able to go to a few sites that I was looking at and actually collect. Unfortunately, COVID got a little bit too crazy at that time, along with the fires. And so that travel aspect kind of got deteriorated within my research. And so I'm kind of just making do with what I have right now, unfortunately, but making the best of it. There is a question in the chat as well from Abe. Are you able to share your herbaria label corrections with the respective herbaria so they can correct them in their database? Yes, that is something that I heavily plan on doing, because I'm hoping that anyone else that plans on using this open access data does not need to chase someone down, so that way they can figure out why something was 530 feet when it was supposed to be meters. So yes, absolutely, I will be doing that. Lyle.
Yeah, I'm wondering if in your herbaria searches, whether you find anything from way back that perhaps has been extirpated.
Sorry, unfortunately, this is going to be really sad, but could you define that term for me?
Extirpated, extinct, no longer exist. Yeah.
That very much potentially can see that. I have not actually gone far enough into any of my analyses to do that. I'm still trying to just go through my data and fix it. So if I were to look at iNaturalist for something that existed perhaps in like the 1850s and it no longer appeared on iNaturalist, okay, well, at least modernly we can tell that it's not presently alive or around at all. I could also do a lit search, but that also becomes quite limited because I do have most of the more recent records, and like the book that I have chosen to use is an older one, meaning that yes, things in here could definitely be extinct, and I definitely would have to go out and grab the next updated manual to see whether or not it truly has been. But even if it was, I think it would still be included in here, which makes it a little bit difficult because I'm not sure that they just eliminate them if they haven't seen them recently and they can just claim that they are fully extinct, perhaps locally, but outside of the range, you might be able to still find them. I don't really know if that answered your question.
Yes, thank you.
Abe and Katrina?
Hi, I'm wondering, great talk. I'm wondering about the resolution you might have on, say, herbaria records from the 80s or prior to like really good GPS. Are you optimistic you're going to have the resolution you need to see in the mountains? A horizontal change of just a few feet could represent, a pretty big vertical change.
Yeah, and so that was something I actually had a question on, and I asked to one of my advisors, and so she recommended creating a digital elevation model and trying to identify if it was township range, how to narrow it down to like an exact locality, because a lot of those actually just didn't come with any elevations, period, because they were older and they didn't directly know exactly where they were. There are a lot of species also that had descriptors, like site descriptors, without township range, elevation, or latitude, longitude. And so those kind of fall into that troublesome category as well, where I'm not fully sure if I will have the resolution. And at that point, I might have to eliminate them just based on what they have presented for me, so I don't confound my data. Did that answer your question?
Yeah, thank you.
Lyle, did you still have a question?
No, sorry.
Any other questions for LiAnn? You can ask what her favorite plants are from the California flora. Anything that you're interested in?
I've got
including...
a question.
Yeah, go ahead.
Yeah. So LiAnn, I came in a little late and maybe I missed this, but I'm wondering about the potential for bias in this research, that the plants you're studying, the plants that you know about, right? And maybe the plants that are most adaptive are the ones that you're most likely to know about. So how could that affect your results, and how would you potentially account for that?
Yeah, so that was definitely a problem that I had. That's also why I chose a much earlier edition or earlier published piece. So this was from 1994. So it's not the earliest that I could have gone, but it was the most clear in terms of what species were present that I could go back to. But I also had that similar train of thought when I was going through. I was like, what if I find something that says it's endemic, but guess what? It's in Nevada too. And so how can I really determine whether or not this species truly is endemic like I say it is? And so that is something I am still working on getting through with my advisor, Mike Williams. We're pretty hopeful that their definition of endemic did not just contribute to the giant renaissance of collection that only occurred in California and not Nevada. And that's the reason we couldn't find anything that would consider it non-endemic. But that's a question I'm still trying to work around, but a very good one.
LiAnn, I assume, sorry, Alan, you had a question? Go ahead.
Oh, I've just wondered why exclude Nevada, because the same mountains, say around Lake Tahoe, extend, the same habitat extends into Nevada, and that teeny bit of Sierras, if it would be very easy for an alpine plant to have its range extended to Nevada, just those few miles. but not the rest of the state. That teeny bit of Nevada shouldn't exclude a plant from the study. That's what it seems like to me. I was born in Reno, so it's close to home for me.
Okay. Yeah. So we were primarily focused on like the Cascade and the Sierra Nevada like intersect and where that kind of touched bases. And so that wasn't necessarily close enough to Nevada. However, we are still going to be taking that into account because I think I did already find a species on iNaturalist that was endemic to California per my study, but also found in Nevada. So that's also another outlier that we might be looking at a little bit closer, particularly if its sample size is large. The study is still changing, unfortunately, but yes, we will be hopefully looking at Nevada as well, should the Cascades and Sierras not really have any index of similarity for me to use.
Well, if a plant population was migrating north from the Sierras, it wouldn't go further into Nevada. It would be going away from Nevada toward following the Cascades. So yeah.
That's what we would definitely hope.
I would hope so.
Yeah, but
northern Nevada is pretty much a desert. So
yeah, but it's quite interesting. The little pocket reservoirs that you'd find these species in, given that like they are niche adapted, and so if there was something available, very high potential you could find it there as well. And that just might be a spot that it decides to chillax in and then go extinct because it's unable to move any further out of that niche.
Okay.
LiAnn, Lyle, you have another question?
Yeah, out of
Yeah,
pure ignorance, I'll ask it anyway. But in these alpine regions, do you have any sense of the preponderance of perennials versus annuals in the alpine regions?
So that's not something I actually ended up looking at. It was something that Mike did propose, but for the realms of my study, it would have unfortunately been a little bit too much. Kind of just like the dispersal barriers, that's something that I'd move on to should my conclusion be that like, yes, they're able to migrate, but like, why are only some able to and not others? That would kind of be something I move into in the PhD if I chose to do that.
Thank you.
When do we expect to see the results?
Ooh, that's a good question. I am supposedly supposed to graduate in spring. I do not think that's going to happen because of COVID. Realistically, I'm hoping for fall, granted that a lot of the analysis goes well, and Ben, who is chair of the department, unfortunately is getting hammered by a lot of problems at the university, COVID related, is able to free up his schedule and get a little bit more time working with me. So until then, I'm hoping for fall tentatively. We will see what happens with that though.
Yeah, I'm hoping we can get her back to tell us the story, how it turns out.
Yeah, you guys are asking really amazing questions. I've presented to my cohort, which is marine based. And so the questions were very much like, wow, what's iNaturalist? But these are amazing and very helpful. And I will be rewatching these just for the questions.
Lyle, go ahead. Lyle, do you have another question, Lyle?
Unmute. So, I've been not being a scientist by any means. I've been kind of curious about how mathematics, statistics, they're essential to these kind of questions. But it sounds like you're kind of learning the statistics along with your studies. Am I correct in that assumption?
Absolutely. When I came in, I had the most basal idea of what an ANOVA was based on just a small class that I took when I was an undergrad. Little did I know that I would be taking multivariate analysis alongside my actual data collection and analysis to kind of start changing gears and actually formulating hypotheses properly. Statistics are insanely important in what I'm doing, and I didn't necessarily realize that until my second year, which is unfortunate. A lot of the first years right now are taking multivariate. And so they're getting a decent idea of what they need for their projects. But retrospectively, I do think that it was better for me to have taken it my second year, because I've realized the mess of data I have and realized the amount of respect I hold for the stats class that I am taking right now. I'm not sure I would have had that respect for it the first year, because I didn't realize how essential it was. But it's key in really understanding what's going on, because you can ask a very basal question and say, I want to compare this landmass and this landmass. But taking multivariate and other statistics courses gets you into that mindset of, you can take those land masses, but you need to understand that those are different sizes. Now you have to make other sizes and shapes. And now you have to have a hypothesis that changes to another question, that changed to another question, that might change the entire framework of your study. And so I think that's really amazing. I kind of went down a rabbit hole there, but I'm taking statistics and I'm kind of overly excited for it right now, clearly.
Excellent. Thank you.
Abe and Katrina.
Well, I applaud your enthusiasm for statistics. I think that's a really important tool in science. Yeah, this is a completely different tack, and I know sometimes it's annoying to have all these other ideas thrown out when you're probably pretty focused on one already. But I was just curious if if any of the species are kind of clonal and might show up on old aerial photography, and maybe you could use. I mean, there's really good, I think, pretty decent at least air photography going back several decades.
Wow. Yeah, I haven't even thought about that.
Because I mean, some high alpine plants, if they're clonal, they might be the only thing in on a scree slope or something like that where there isn't much other vegetation. And with a little, I mean, it would probably take some ground truthing. It'd be really fun ground truthing, but you might be able to track a clonal plant as it moves up a slope or something like that in air photos.
That's really interesting. I hadn't thought about that. That might be kind of difficult, because I could be like, okay, in this general vicinity, we found a cluster of colonial plants. And then now I need to go see if a naturalist, someone is able to hike in that exact location so they can tell me what that species is, if I'm not able to aerially see what species that actually necessarily is. That's an amazing idea that I haven't thought of. Kind of sounds like tracking permafrost almost. But yeah, that's really cool. I'll have to bring that up to Mike. Not necessarily me at the moment, but Mike would love to hear more about that.
Yeah, and I mean, it's quite possible that that a lot of these herbaceous flowers just aren't going to be robust enough or dense enough to show up. But you know, something like a Krumholtz tree would show up, I'm sure.
Mm-hmm, for sure. Eric, Abe, did you have any more, or Katrina?
No, thank you. You could turn.
I have a question even more tangential than Abe's. It just coincidentally lately I've been really curious about how this area was revegetated after the glaciers retreated, which was about 10,000 years ago. But I've just been really curious about how quickly that occurred, or you know, what phases that went through. Just wonder if you happen to know anything about that, you could fill us in.
Unfortunately, that is not something I have any capabilities of defining. Eric Deshane is a master in that. He's really well versed in just anything related to glacial recessions and what pops up after. He actually has another master student right now, Miles Berkey, who would be amazing if you guys ever decided to bring him here to talk. He studies bryophytes, and so he's more well versed in like that kind of mojo.
Abe, do you have some insight on that question? Perhaps general patterns of glaciation and.
Sorry, I was thinking about other things.
Yeah, you've got your hands full with a cutie. Oh my! Oh, everybody, that's Abe and Katrina's little baby boy. How old now? Just a few weeks.
Yep, 8 weeks old. Oh, this is Forrest. You enjoyed your
so cute. Thank you for sharing. So, any thoughts, Abe?
About plants.
A glaciation of the pattern of glaciation in the and how it affects California endemics. How they may have been affected.
Oh, I just don't really know the flora down there.
Yeah. Okay. Sorry. Perhaps Eric, that was your question, right? So Mark is up next. Go ahead, Mark.
Yeah, so you mentioned iNaturalist a number of times, and I've looked at records on iNaturalist trying to locate specimens of plants to go out and find in the field, and photograph. And some of the records are really good quality, and some are, well, a little sketchy, and you're not sure whether the identification is correct. So I wondered if you have any thoughts on working with those, that crowdsourced data compared to the pretty rigorously checked herbarium specimens. I think if I look at an herbarium specimen, I can be pretty confident that somebody who knows their stuff has looked at it and said, yes, this is Polemonium pulchurinum variety shastensis, as opposed to variety something or other.
Yeah.
The thoughts on that?
So I use the research grade only from iNaturalist just because I physically couldn't go out and check and confirm anything. And visually, for me personally, I've gone as far as taking systematics and doing a few plant surveys at the Hawaii Institute of Marine Biology, but I'm by no means an expert. So I would definitely be bringing in Mike Williams for that. That's his business, but I really didn't work with anything past research grade. If it didn't say research grade, I tended to avoid it, just again, in case of error. But if there was only one retrieved hit or image and it was non-research grade, shaky, I just didn't look at it, unfortunately, because I really didn't want to introduce that error. Did that answer your question?
Yeah, and that's pretty much what I was doing as well. If it hadn't been verified by somebody, which is really what research grade on iNaturalist is at least one other person that says, yep, you're right.
Mm-hmm. Lyle, do you have another question?
Oh, well, not so much a question, but just a shout out to one of our chapter members, Barry Wendling, who you might be familiar with. And in terms of refugia and, glacial, refugia and things like that. I don't know if you're
Barry
familiar
here?
with it. I don't think Barry is here.
I checked to see if he's here because yes, you're totally right. He would be.
But I know he's worked closely with Eric Deshane, and gone on some just mind-blowing expeditions in Siberia and God knows where. So, yeah.
Definitely. I haven't seen him around campus much recently. I think just because of COVID, it's been a little bit offset, but could definitely ask that question or run it by him if I do see him on campus again, potentially in the herbarium. Any more questions?
Well, LiAnn, let me thank you. It's been wonderful. Thank you for sharing your project, and we hope to hear your results when you have them. And that'll be another exciting story for you to tell us about how things turned out. So thank you very much.
Thanks for having me, and thanks for all the amazing questions. You guys really took my brain for a spin today, which I needed. So amazing. Thank you so much for giving me this opportunity.
We can stop the recording now.